Hello, and welcome to this talk on systematic variety naming in cannabis, why there's a necessity for it, and what some approaches to doing that might be. I'm Dr. John Brunstein from Segra International. Some quick disclaimers there. Segra is a Canadian-based cannabis tissue culture nursery business. We grow plants at industrial scale for LPs. We can also do preservation and regeneration services by tissue culture for people. And I oversee a genetics lab which performs various tests from everything from pathogen detection to what I'm going to talk about today, which is genetic identification of plants. We do this both internally and as an external fee for service. There are other methods other than the way we do this to do this approach, and I'm going to try to be as even-handed as possible and discuss some of them as well. One other disclaimer before I move on is that the term strain technically should be used for microorganisms, so I'm going to use the terms variety or cultivar throughout this talk, except in cases where I'm directly quoting. Customers buy cannabis based on name. In both the old and newer versions of Leafly, a common website that people go to for looking up cannabis, it ties variety names to crowdsourced reactions. So there's a linkage between name and effect. Scientific data is similarly paired to names. GenBank, last time I checked, had nearly 90,000 cannabis entries with self-identified names. We see names used in biomedical publications as well, so this is a prevalent thing to do. Names have meanings in other organisms. We can tell one kind of dog from another. We can tell one kind of grape from another, and the type of wine that comes out of that grape can be quite different between a Petite Sirah and a Muscat, and people pay differently depending on what it is. Well, could we also do that in a green plant, dioecious, where the female inflorescences contain some sort of terpenoid compounds that we want to use at an industrial scale? Such a plant could be hops, and the answer there is absolutely yes. Hops have distinct varieties. Those varieties have distinct reproducible characteristics, and if you buy a Nugget or a Citra or a Bravo hops, you know what its characteristics are going to be. Cannabis has a lot of variety names as well, but most of these are self-reported. One clone could be sold under more than one name, or you could have different clones. Sometimes more popular name, a lot of things could get sold as that. Varieties named for a cross are frequently named by conjunction of the parental variety names. So two different breeders doing a similar cross could produce an offspring product with the same name, but these are not identical. They're siblings, not twins. Accidental or willful mislabeling, either way, sometime in the past, can of course propagate forward. So even materials now in robust track and trace systems, depending on where they've come from, may not always be reliably named. Back in 2017, we wanted to know how many names there were, so we did a review of online dried flower for sale in a couple of Canadian and American jurisdictions. We looked at a total of 2,739 items, and we found 1,263 names. But 9 names alone were already 10% of the total population. At the other end of the scale, 845 names only occurred once. It's unlikely all of those were actually unique. So the question is, do these variety names have a reliable meaning in cannabis? Well, how can we tell? DNA fingerprinting is an obvious approach. No, plants don't have fingers, but a fingerprint in this context is a reproducible biometric mark of identity. Like a real fingerprint, which doesn't tell you if the person has blue eyes or blonde hair, a DNA fingerprint may not necessarily disclose physical phenotypic parameters, and we'll talk a little bit more about that later. The method we use in our laboratory is VNTR profiling. This is based on a small number of highly size variable loci, and it's the method used in a lot of human forensics because it's reliable, it's robust, it has small data sets, and it's easily analyzed for identity and relatedness. Luckily, suitable loci already exist in cannabis and are well understood in the literature. You don't have to be a specialist to understand what the data looks like. The blue peaks here are measurements of a particular size of loci in 3 different cannabis samples, and this one has 2 at 90 and 101 base pairs, this one has 2 at 90 and 96 base pairs, and this one is a homozygous, or possibly hemizygous, 90-90. And even to the non-specialist, this doesn't look like this, doesn't look like this. Those are different clones. We take and now multiplex this across multiple loci. In our case, we do 12 different loci. We generate these numbers in order, 24 numbers. This is something we call a VNTR vector. Vectors are identical between identical clones, and they're different between different materials. The probability of identity, or the chance that any such strings of numbers are the same between two things that aren't related, is vanishingly small. Somewhere between 1 in 14 million and 1 in 10 to the 49th at the two extremes, theoretically, for the sample we do. And in reality, for an average specimen, if you look at the math, it's about 1 in 100 million chance of a random match. This sort of data can beget phylogenetic trees as one way of visualizing data. And of course, tree topologies are models. The actual shape of the tree is not always correct, but absolute distances are true. You can easily tell things that are clonal from things that are clones to near neighbors, and you can tell things that are near genetically versus far genetically. So let's go back to our question. Are cannabis varieties named reliably? Well, sometimes yes. So these two shishka berries are clonal, those two shishka berries are clonal, and these two and those two really, on the scale of the entire phylogenetic tree, are very close to each other. These two things are both called cheese of some variety. They're not identical, but they're really close. These two things called green crack down here were, in fact, clonal. But often, no. You find examples of one name, different genotype, and I won't go into details, but in this case, chemotype as well. All of the things shown there on the tree, the green star is 3 examples, closely clustered of something called Girl Scout cookies, and the red stars are each examples of something else called that. They're not all Girl Scout cookies. Blue Dream was the single most common name in our sample. It was 2.1% of the entire population we saw for sale. 3 different things here called Blue Dream, not close to each other at all. I don't know if any of them are the right Blue Dream, but they're certainly not all the right Blue Dream. You can see the inverse of that. In this case, we have one particular clone here on this big isogenic line. We've seen at this point a total of 22 names applied to that clone. If we do replicate sampling of legal stream material, we took 10 dried flower products purchased in the BC government chain, each tied to a producer and a product name. We fingerprinted them. We waited 2 to 3 months and we repurchased the same product, same LP, again did a fingerprint, and we compared. Well, 5 out of the 10 samples turned out to be the same lot number when we got them again, so mostly we learned that those products didn't sell extremely well, but all 5 of those scored isogenic to prior sample. The other 5, however, were new lot numbers. 3 of those 5 were isogenic. 1 wasn't isogenic to its prior sample, but was close, but 1 out of the 5 was nowhere even close to its previous material. What if we now do replicate sampling on single vials? Well, we tried that too. BC government legal supply chain, 1 gram samples of dried flowers sold as a single product variety name. We took multiple samples from distinct buds in the container, and we VNTR'd them. What did we find? Well, out of 12 samples tested, most of the time, 1 vial has 1 genotype, but 1 vial detected with 3 different genotypes, 1 vial detected with 4 different genotypes, and within vials, these genotypes were not all near neighbors. They were quite separate genetically. Notably, both of these vials turned out to be from a single licensed producer. I'll let you draw your own conclusions from what that might mean. What are the costs of inaccurate naming? Well, on a recreational side, it impacts branding and customer expectations if you don't get the same thing every time when you buy the same label. On the medical side, it's maybe more serious because the variety and the dose titration is very personal to get a good therapeutic window. One could imagine seller liability if a person is taking cannabis for, let's say, chronic pain relief, doesn't normally get drowsiness, gets something with the name they expect, takes it, gets behind the wheel of a car, and it's the wrong thing. They do get drowsy, they get into an accident. Imagine what could happen there. On the research side, medical research and breeding programs are both negatively impacted if we can't reliably share data between different places and different studies. All of these suffer unless variety names are applied consistently. Really, why should dogs or hops or grapes or roses have reliable breed or varietal names but not cannabis? So what makes a variety? Well, it's a commonality of phenotypic traits of importance. Both of these in this picture we would call Dalmatians because a breeders association has decided that brown versus black spots is not a significant variation, but that's a personal choice. And this sort of choice has to be made by type or reference exemplars by some body of authority. And there needs to be an allowance for a certain range of phenotypic diversity, brown versus black here, arising from nurture, or in this case, a limited genetic diversity under a single appellation. Basically, genetics provides the potential for the phenotype. Nurture can fine-tune the expressed phenotype, but people have to determine which phenotypic variations are significant enough to warrant a new name. How do we measure genetic diversity? Well, there's multiple methods available. They're all molecular biology based. Rapid and ICR are very low tech. VNTR, the method we use here. There's also things like single nucleotide polymorphism or SNP panels and genotyping by sequencing. Each of these has pros and cons. In particular, I would suggest, though, that SNP panels and genotyping by sequencing have significant capacity to disclose linked phenotypic traits. In other words, unlike a fingerprint in our analogy, SNPs or GBS would tell you that the person had blue eyes and blonde hair. Or in this case, they might disclose detailed aspects of the phenotype, the chemotype, the disease resistance in a plant. A breeder might not want to be disclosing that. They just want a way to get a handle on identity, and that's where we think VNTR kind of hits the sweet spot. Multiple methods can, of course, coexist if they share an exemplar library. How do we interpret genetic diversity? Well, phylogenetic trees are one way, but what about principal component analysis? PCA is hard to visualize meaningfully in 2 or 3 dimensions because it's multidimensional, but it does allow for computation of point-to-point distances between any 2 genetic points, and it allows for relatively easy computation of self-assorting clusters or groups with centroids, so a cluster of things that maybe you want to apply a name to. Alternatively, you can see this with a range of genetically distinct samples that you have said, I know what the name of this, this, and this are, and now you have specific points and you accept things around that point. Well, accepted by who, again, goes back to this need for a buddy. The other side, of course, is chemotype. Now, chemotype can also be done by multivariate chemotypic analysis. For cannabis, you likely want to use both GCMS and HPLC because of the range of volatility and polarity of the molecules you're looking at, but the results are equally amenable to PCA, and again, you get this ability to have self-associating clusters and centroids, or you can have a body provide reference materials and make these centers and decide what variations are acceptable. In fact, there's a group out of Holland that's done this kind of analysis, Hazecamp's group, one of their papers, figure from that is shown here, and they claim that chemotypically there's only really 8 cannabis chemotypes. Well, moving forward, what should we do? I think the question here is what constitutes a suitable body? Probably, I would suggest a group of recognized authorities, some long-term breeders, seed banks, members likely spread internationally. What do they need to define? Well, type specimens for extant popular variety names, some sort of methods or metrics for both genetic fingerprint and chemotypic analysis, and then finally inclusion or exclusion criteria based on both genotypic and chemotypic similarity to a reference centroid for a variety. If you're too different genetically or too different chemically, you're not the same variety. Well, for new varieties, this is probably not too hard to handle because there could be some way to submit a sample for genetic fingerprinting and chemotypic signature to the body. And there could be issuance of some sort of recognition of a variety name, some sort of certificate. Extant varieties, well, some have traceable original clones, and you can go back and do this. If not, I don't know, maybe a majority rules from members of this body or panel. What about other options? I think this is a topic for discussion. Too long to get into in this talk, but hopefully this stirs some thought about that. What are the challenges and benefits of doing this? Well, the challenges primarily, I think, are that some producers might need to rename a current product to harmonize with the rules. But the benefits for breeders includes protection of novel varieties. For consumers, medical and recreational, improved uniformity of product. For researchers, improved capacity to find correlation between variety and effects. And for producers, an ability to market based on a provable uniqueness. Hey, our product is different than everyone else, and here's the proof. Has this been done before? I won't go into the whole story, but in human immunology, it's been done. People all over the world were working on molecules. They were naming them, whatever it was in their own lab, and using antibodies to identify them. Eventually, they got together, pooled their antibodies, and then found the ones that all bound the same thing and called those cluster designation 1, 2, 3, 4, now known as CD4, CD8, CD22. These are, you can pick up a newspaper and see these terms now. It is possible to get everybody to rename in the same fashion. Final thoughts, my last couple of seconds here. Is there an alternative? Yes. We can all choose to simultaneously believe multiple mutually contradictory things, which are cannabis variety names as they currently exist. If we do that, I'm going to suggest we all need to get an electric monk. And for those of you who don't know what that is, this is a handy device suggested by Douglas Adams. And to quote partially here, electric monks believe things for you, thus saving you what is becoming an increasingly onerous task. And with that, I'd like to give my acknowledgments. Obviously, the people at Segra, special thanks to Jerian, Ying, May, Manmeet, and Paramjeet in my laboratory, and you, the audience, for your attention. And with that, I will be happy to take questions however it is we're going to do that in this format.